We're going to take a little bit closer look at the sliding filament mechanism. I realize that you've already seen a video on this, but I wanted to talk about it just a little bit more. So the basic idea behind the sliding filament mechanism is that myosin pulls on actin. So you can see here a little clip from the video that you've watched. Myosin pulls on actin. This causes the thin filament to slide inward. And so basically the thin filaments, because they're attached to the Z discs, when they're pulled in towards the M line of the sarcomere, that's going to pull the Z discs in towards the M line. So basically, the sarcomere is going to shorten. So as you can see here, those Z discs actually get moved towards one another, and the sarcomere is going to shorten. So again, I want you to remember that the thick filaments have a direct connection with the M line, that the thin filaments have a direct connection with the Z discs. And so basically, when those thick filaments pull on the thin filaments, you're going to see the Z discs move towards the end line, and the sarcomere shortens. Now, as one sarcomere shortens, it doesn't just shorten in isolation. All of the sarcomeres are going to shorten at essentially the same time. And so, with the sarcomere shortening at the same time, the entire muscle fiber shortens, and as a consequence, the entire muscle is going to contract. Okay, now the next thing that I want you to notice here are the changes in the I band and the H zone as the muscle contracts. So let's get reset a little bit here. Okay, so it's, ironically enough, it's going to be, I think, a little bit easier following the micrograph on the right, so the pictures on the right, than it is the diagrams on the left. But let's get oriented anyway. Remember, the M line is the middle of the sarcomere. If you want a cheap way to remember it, you can think of the M line as the midline of the sarcomere. Okay? The H zone is the area where you have thick filaments only, so there's no overlap in this area. The I band is the area of the sarcomere where there's thin filaments only, so again, there's no overlap. So if you take a look at the top diagram, you see the muscle in relaxed shape, okay? And so basically the sarcomere is about as long as it's going to be. You've got a substantial H zone. You've got a substantial I band because there's not a lot of overlap at this point. Now as the muscle begins to contract, you can see moving from the first to the second diagram that the H zone has narrowed down. You can also see that the I bands have narrowed down, and look at the position of the Z discs. They've actually moved in towards the M line. Also, I want you to notice that the very darkest portion of the sarcomere has gotten thicker, so take a look at that. As you move from partially contracted muscle to maximally contracted muscle, the H zone almost disappears. It's because the thick filaments have dragged those thin filaments in so close to the M line that there's hardly any H zone left at all. I also want you to notice that the I bands have also contracted down really short. And again, the reason why is because there's simply a greater amount of overlap between thick and thin filaments. So the I band, the H zone, they're going to change as the muscle contracts. We're going to take another look at the contraction cycle here. I realize that you've seen this on the video, but there are some things that I wanted to emphasize that the video didn't. So we're going to take a look at this step by step. So in the first step, myosin heads hydrolyze ATP. And this is the first piece of information that I wanted to give you. That myosin doesn't just act as a contractile protein. Myosin also acts as an enzyme that breaks ATP. In other words, it is an ATPase. I want you to keep this fact in mind for a future video. So now myosin has bound to ATP. It has broken ATP into ADP and phosphate with a phosphate present on myosin. Then this means now that myosin has the ability to bind to actin. Without a phosphate present on the myosin, it does not have the ability to form a cross bridge. So now we've got the phosphate there, and myosin can form its cross bridge with actin. So myosin binds to actin. Now, ironically enough, in the next step, even though phosphate was a requirement for binding. After the binding occurs, the phosphate will dissociate or leave the myosin. And as you know, myosin being a protein, if something binds to a protein or unbinds from a protein, the protein is going to change shape. And in this case, when phosphate leaves, then the myosin changes to a shape that will give it a stronger cross bridge. Think of it as sort of strengthening its grip on the actin. So the phosphate dissociates from myosin. This strengthens the cross bridge. In the next step, another unbinding occurs. ADP unbinds from the myosin. This causes a shape change in the myosin. And so what's going to happen here is the power stroke, in which the myosin-actin cross bridges get moved towards the M line or the center of the sarcomere. Now, once that power stroke has occurred, the next thing that happens is that ATP binds to myosin. Remember that myosin has the ability to bind to ATP. But when ATP binds, the myosin is actually going to change shape again. When ATP binds to myosin, that shape change will cause the myosin to change shape such that the cross bridges break. In other words, ATP must be present in order for cross-bridging to occur. We'll examine the implications of this a little bit later on. The next thing that I want to do is talk about excitation-contraction coupling. And so basically, the idea here is that a muscle action potential connects the excitation of the membrane, which is the excitation means that there's an action potential moving along the membrane. It connects excitation with contraction. So let's take a look at what's going on when the calcium release channels are closed. So moving in on the sarcoplasmic reticulum, you can see here that these calcium release channels are closed. Now that blue exit you see at the bottom, this represents a calcium pump. Now those things don't ever turn off. So the calcium pumps are always trying to move calcium into the sarcoplasmic reticulum. So at this point, there's a massive concentration of calcium within the sarcoplasmic reticulum, about 10,000 times what it is in the rest of the sarcoplasm. So here, these release channels are closed, pumps working, lots of calcium in the sarcoplasmic reticulum, very little of it elsewhere. Okay, let's take a look at the configuration of the thin filaments. Okay. Now, one thing that I want to remind you of is in skeletal muscle, the thick filaments are always ready for action. They bind to ATP, they break ATP, they are ready to cross bridge. And so what's the deciding factor here whether contraction occurs? The deciding factor is whether actin is also ready for business. And in this case, actin is not ready for business because tropomyosin, which is the brown-looking filament, tropomyosin is blocking the binding sites on the actin. So actin does not have the ability at all to bind to myosin. It is being regulated at this point. That's the reason why troponin and tropomyosin are called regulatory proteins. So again, the troponin is holding the tropomyosin over the binding sites on actin. So cross-bridging at this point cannot occur. Now what happens when the calcium release channels actually open? Let's zoom in on the sarcoplasmic reticulum again. Now again, notice that those calcium pumps, they're still going, but when those calcium release channels open, it completely overwhelms the ability of the calcium pumps. It's like a flood of calcium moving out into the sarcoplasmic reticulum. Now the reason why is because one thing that we learned about diffusion in lab and also in lecture, it was the greater the concentration gradient, in other words, the greater the difference in concentration between one area and another, the faster that diffusion will happen. So with a 10,000 to 1 gradient between the inside and the outside of the sarcoplasmic reticulum, when these channels open, calcium will flood the entire sarcoplasm immediately, or essentially immediately. Like I said, it completely overwhelms the ability of the pumps to adjust to this. Bang! Calcium leaves the sarcoplasmic reticulum. In this fourth diagram, you can see that the calcium has now bound to the troponin. So when calcium binds to troponin, it changes the shape of the troponin and it moves the tropomyosin away from the actin binding sites. So now at this point, with the binding sites on the actin uncovered, that means that actin is now open for business and cross bridges can occur. So I want to also talk about a little bit of terminology here. You see that it says myosin binding site on actin. Okay, that refers to the actin molecule. So the actin molecule has a small portion that can bind to myosin. This is the myosin binding site, and it's that site that is actually covered by tropomyosin when the muscle is relaxed. Now, myosin also has an actin binding site, but that actin binding site is always open, and in the case of skeletal muscle, myosin is always ready to go.